Welcome back. So in today's video lecture, we will look at the demonstration of optical tensiometer. So in last video lecture, we looked at the force tensiometer. So as you know, force tensiometer is used to measure the interfacial tension of various plates, okay, using the linear plate method or Donner-Ring method. So today we are going to look at optical tensiometer, which is a complementary technique to force tensiometer. Okay, and so sometimes optical tensiometer is also called as goniometer. So in Greek language, gonio means angle and meter means metric. So collectively it is called as contact angle meter. Okay, so one can say goniometer also as or also optical tensiometer. One can call it as optical tensiometer also or goniometer also. Right. So in today's lecture, we will look at the overview of the goniometer and followed by the demonstration by our one of my PhD students. Okay, let's begin. Yeah, so as just now described, so optical tensiometer can also be used, right, in place of force tensiometer to determine the surface tension. So not only surface tension, you can also perform, contact angle measurement using Cecil drop method. In addition to the surface tension using pendant drop method. Okay. Sometimes bubble pressure tensiometer is also used to determine the surface tension. Okay. So that is nothing but bubble pressure tensiometer. Okay. Right. So essentially it consists of following components. It has got a camera, okay, which is fit at the one end, and you have the dispenser, okay, usually a syringe filled with liquid, of interest, and then so using that one can perform pendant drop as well as Cecil drop method. Then you have got sample stage. So this sample stage can be moved up and down manually or automatically. Okay. Then you have got a light source. So basically this light source will will help you to track the to capture the droplet image, right, on the sample stage. Okay. All right. So, what are the main advantages of this method and how it is usually functioning? So, one of the main advantages, there are several advantages of using optical tensiometer. You can see that this is, often done in non-contact mode. Okay, which means that because you basically this experiment and this device help you determine the surface tension or contact angle using imaging using imaging method. You can say that this is also a non-destructive method, and unlike your force tensiometer, it doesn't require a huge sample volume. So all you need is just a few microliter sample, right, and easy to handle. Unlike the force tensiometer, you won't need to really worry about the probe which we use. Let's say linear plate or Donner-Ring. Okay, both are made up of platinum or special alloyed material. Okay, and also often in force tensiometer, we need to ensure cleaning, right, after every cycle. But in this case, you don't really need to, worry about those factors, right? And there are certain limitations like because this method is image-based technique, so one would need to worry about the sensitivity to resolution, right? While capturing the image, right? And also setup complexity, which means that every time one has to perform the calibration, because this is based on the, so there is something called baseline correction, okay, and there is something called calibration, height width calibration. So this needs to be performed every time when we use this goniometer, right? So this is one of these are a few limitations of this technique. Okay, so how it works? So it has got a camera and it has got a light source, and you have a needle filled with liquid of your interest. So if you want to perform the pendant drop method, one has to slowly generate a droplet. Okay, so because when you generate droplet, there are two forces acting on this droplet. One is the gravitational force, another one is the surface tension force. Because the liquid wants to wet the surface, it will be strongly held by the needle at the tip of the needle. On the other hand, there is also gravity acting downward. So these counteracting forces make the droplet make the droplet little bit distorted. So they will not display a spherical. So there will not be. So they will display like a pendant. They display. The droplet will be in a pendant shape, right? Mostly, right? Because of these forces acting on the liquid. Because this is not exactly spherical, so one cannot use the constituent relation as such. So one needs to modify according to the shape factor. So one has to use the shape factor here. And the other one that you can easily perform is Hessian drop method. In this, you will have to, the droplet has to be placed on the surface of your interest, and then the side view of the droplet will be captured using the camera at the fitted at the side, right? And then performing the baseline correction and using the curve fitting. There are several curve fitting method used, okay? So based on curve fitting, one can calculate or determine the contact angle, right? Yeah. So you can see here, there's a syringe kept over here. So this can be operated manually also or automatically, okay? In some of the latest optical tensiometer, you also have an additional module called pulsating module, using which we can actually generate surface pressure versus area data, which will help us calculate perform the interfacial rheology analysis, based on interfacial rheology. So which means that one can find out what is the G prime, J double prime, okay, of the colloidal particles, absorbed at the interface, right? So interfacial rheology can be performed using this module. So some of the latest technique available today, provides these facilities, okay, right? These modules. So next we will look at see how this pendant drop method is basically carried out. So as I told, this shape factor beta is very important if you want to determine the surface tension. So that is readily determined using the various geometric parameter, okay, which is shown in this pendant drop schematic. Once the beta is calculated, once software calculates the beta, based on this pendant shape, based on these geometric factors, you can determine surface tension easily from this relation, right? So in addition to it, we also have what is known as bubble pressure tensiometer. We also looked at a bubble pressure method when we described about the various measurement techniques. Okay, so this is one of the measurement techniques that we already have. We already covered, right? When we discussed about the contact angle measurement techniques, right? Right, or surface tension measurement techniques. So it uses. This device help us determine perform the dynamic surface tension measurements. Okay, so by calculating the maximum internal pressure of a gas bubble, we can calculate the surface tension using this relation. Okay, all right. So this schematic we already have seen during one of our lectures in module 2. Okay. So now we will stop here, but one of my students will explain you about the optical tensiometer, okay, by demonstrating using some liquid samples. Okay, thank you.
Welcome back. Today we will see the working of the goniometer, or you can see the optical tensiometer. This is our optical tensiometer made by Baldwin Scientific Attention Theta Flags. There are mainly 3 components, or we can say the basic components. As you can see here, one is the camera attached to it, or in parallel there is light source attached to this. Another component is the stage, where you have to put your sample, and a hanging cantilever where you will hang your syringe. To adjust the tilting of the camera, we have a adjusting facility. By rolling this ruler, you can access the tilt angle of the camera, especially for sessile drop measurement, when you are going to measure the contact angle using the sessile drop measurement, right? So in that case, we have to keep this camera at -2 degree. You can actually see the surface and the formed droplet on the surface. With the help of that, you can calculate the contact angle of the system. Also, this stage can move manually like this. You can move it up and down it manually. You can adjust the z-axis, y-axis, and the x-axis one by one using these tools, which are readily available in the system. You can also adjust the height of the cantilever beam for hanging the syringe. Now, let us move forward to see the software interface for the system. As you can see here, this is the software in one attention, where after clicking, you will open it. This is the main software window. You can choose test accordingly, whether you want to do sessile drop, bass sessile drop, pendant drop, meniscus, dynamic contact angle, pulsating drop. Here I am going to show the sessile drop for the measurement of the contact angle of a surface using water. After clicking the sessile drop application, you will see this type of interface. In this system, you will get different type of needles that can be attached to the syringe. Each and every needle has a different edge and diameter that you can choose according to your sample. Suppose you have highly viscous or less viscous, you can choose accordingly. This depends on the applications. You can see here, this is a smaller one with a lower gauge needle. The minimum size is like this, as you can see here. We have another one that is little bigger than this. This is the bigger one with the bigger diameter needle. Apart from this, if someone wants to measure the interfacial tension of an oil in water system, as you know, the density of oil is less than water. If you put the droplet inside water, it will float upward towards the interface. In this case, this J-type needle, or you can say U-hook shaped needle, will be used to measure the interfacial tension. All these needles can be set in different syringe available. Here I am showing you the manual operating syringe. This is made by Hamilton. This also can be used with the help of a syringe pump. To use this syringe, just rotate it manually. By rotating, you can open it and close it. You can also see here. Or we have one more syringe needle. To use this syringe, just rotate it manually. By rotating, you can open and close it, you can see here, this moves by rotating it anti-clockwise or clockwise. This will drop liquid. Now let's see how we measure the contact angle of our glass. Right here, I will show you the contact angle measurement of a glass slide to understand whether the particular glass has a hydrophobic or a hydrophilic nature. I am using normal glass slide available for microscope purposes with the help of water to determine the hydrophobicity or hydrophilicity of the glass slide. Remember, before doing any experiment, whether it is for contact angle, interfacial tension or surface tension, we must calibrate the system. For calibration, I am using this iron ball. Just keep this ball in the middle of the stage here. You can see here, I am keeping this. Now you have to see with the help of the camera. Remember, for calibration, as I told you, for drop measurements, we have to maintain the tilt of our camera at -2. Now we will rotate at -2. By rotating the roller clockwise, you can change the tilt angle of your camera. I'm setting this at -2. As you can see here, this is the scale. This is the scale that shows the tilt of your camera. So here, the scale is at -2. Now our concern is set at a -2 degree tilt angle. Now let us look at this software to see how this ideal setup looks and how to calculate our system. As you can see here, our stage is not visible. Now by changing the z-axis of the stage, I am trying to adjust the ball within the frame. You can change the minor focus in your camera to position this ball in the middle of the window. Now by adjusting the minor focus, you can properly focus on the ball. Once the focus is adjusted, the entire ball will be clearly visible. Now, after focusing on the ball on the stage, you can see that our ball is now properly visible in the system. Remember, calibration is complete when the contact angle formed by the ball is showing 148 point something. With respect to its radius on the surface, your system will be calibrated. You can also change the camera parameters from here. By clicking the parameter button, a window will pop up. You can adjust the exposure, gain, gamma, light intensity. You can also command the system to auto adjust the camera parameter. So I am clicking on the auto adjust option. Now you can see that it is auto adjusting the light exposure, gamma and everything else. Now our system is completely focused and the camera parameters are fully set. Now click on the calibration. Now remember, we are using a ball with a diameter of 4 mm, which is a standard one provided by the system. You can also use needle calibration if you are using a different conditions. Now by clicking the calibration, you can see that the system is showing is calibrated. Now let us move towards the contact angle measurement using the side drop measurement. Now you can see here, this is the manual syringe I am going to use. I am going to use, this is the gauze needle for this manual settings. Now let us see how to refill our syringe. By dipping the needle into the water, I am rotating the screw to fill the water into the syringe. As you can see here, we have sufficient water right now for the cell drop measurement. Now you can hang your syringe like this on the cantilever beam. I'm going to use a glass slide. As you can see here, this is the standard microscopic glass slide. You can use your sample as well. Remember, for cell drop measurement especially, try to use the flattest surface possible so that you can measure the contact angle accurately. Place the glass slide on the stage. Now our glass slide is steady on the stage. By adjusting the ZX position of the stage, I am aligning the height of the glass slide so that it is easily visible in the software window. You can also adjust the minor focus here by using the focusing screw on the camera. You can reset and fine tune the focus as needed using the adjustment. Let us see how glass slide looks under the software window. Now we can see the surface of our glass slide. This is how it looks in the software window when viewed through the camera. Currently, the distance between our substrate or stage and the needle is very high, you can adjust the z-axis using this control. As you can see here, this is moving downward and this is moving upward. You can also adjust the x and y direction of the syringe using this screw so that the droplet will be positioned in the middle of the camera view. Once aligned, let's proceed to sample the droplet. Now, let's put the droplet on the substrate. By rotating the screw slowly, you can release the droplet. As you can see here, the droplet is forming and has already been placed on the substrate. The droplet can now be observed in the software window. This is how the droplet looks in the software window. Before starting the analysis, we need to input a few settings. Let me show you. Go to the recipe. First, we will change the experiment name. That is the demo one. We will put. If you want to autosave your data, enable autosave options. You can make the data private as well. Enable live analysis if you want to see the results in real time. You can also choose the method for contact angle measurement, such as contact angle circle, polynomial, Young Laplace. For this demonstration, I am using the Laplace method. Now, let's define the baseline. And the blue line in the window represent the baseline. You can also choose to set the baseline automatically, manually, or circular automatic. I will be setting the baseline manually. Adjust the baseline according to the surface. Like this, you can adjust it. The baseline represents the flat surface of your substrate. Okay. If the surface is uneven or has a plateau in the certain areas, you can manually adjust the baseline by clicking the orange spot and dragging them as needed. To reset the baseline, double click the orange spots and the baseline will be straightened. Next, select the face material. That is light face and heavy face. Here, heavy face is water that we are using here. Right. After configuring the setting, perform a minor focus adjustment to make the droplet appear accurately on the surface. You can also fine-tune the camera parameter manually or use the auto adjust feature to optimize setting like exposure. You can see here the changes and gamma. Right. Now everything is set. Click on the start to begin the measurement. The live analysis has started. Once the contact angle is calculated, you can stop the measurement. The software will display the measured contact angle on the substrate. By clicking on the Analyze All, you can calculate the average contact angle. Now, in the statistical panel, you can see the results, including mean contact angle, this one, standard deviation, that is 0.06, minimum value, and the maximum value. The results is also visible on the left panel of the screen. Now, to save the data, click on the data, go to the Export All, a pop-up window will appear. Choose the desired format, CSV, Excel, and text, and specify the file name. I am writing here demo 2 in the desktop. Your data is now saved and ready for the further analysis. To save the image of the droplet for documentation, right click on the image and choose the save option. So you can see the pictures in two different modes, or you can also copy it. The native mode is when there is no contact angle shown on the droplet. In the overlay mode, the contact angle will be visible. So I'm saving it in a native mode first. Nope. Okay. Now it is asking in which format: BMP, JPG, PNG. or other format will also come up. I am giving it in the name image 123 and saving it somewhere on the desktop. Okay, right now if you want to measure it or keep it with the contact angle notations, just click it at whatever point you want, or you can analyze all again so that it will give you the average contact angle. Now I can save it and should be the overlay. To see this image, we can check. This is our image with overlay. And this is without overlay. So this image can be used for the further application purposes. Now let's move forward to the pen and drop measurement. As I told you, from application window, just click on the pen and drop. You can see this is how our system and stage looks in the digital position. Now remember, first you have to focus your needle properly so that whatever droplet you form is visible. And you can also check or remove your stage position so that it does not show any type of attraction with the stage. Again, in this first, we have to make a recipe. First, you have to give an experiment name. That is, I'm giving T1. And the same auto-save option. If you want to make it private, you can do it. Lab analysis is also available. Choose the one you want to use. For analysis mode, I am using here the Laplace mode. In Laplace mode. Next, you can use auto baseline. So it will show the auto baseline. If you remove it, it will show the manually. Let's this, you can make the baseline properly. Okay. Remember, this line will represent the flat edges of your needle. Also, if you want to use slip by condition, it will change automatically. You can use evaporation compensation so that if some evaporation is taking place, it can prevent it. Right. You can change the image position as well. If you want to record for a longer time, you can change it from here. I'm just recording it for a 10 second. If you have a syringe pump with this, you can change the setting for the syringe pump as well. Change parameter like the light phase or the heavy phase. Now let's move forward to measure the pen and drop. Now our recipe is ready. I already named as T1. Like I showed you initially in the SSL drop, in the same manner you have to make a drop. This is how your drop looks like in the camera. After adjusting the focus on its edges a little more for clarity, we will do the auto adjust here. Now our droplet is ready. As you can see here, this is giving us a result 72.8. This is the blue line for the contact angle. Now click on start to see the interfacial tension. This is now recording the interfacial tension of the system. Go to analysis and choose your experiment. From analyzer all, you can analyze your measured interfacial tension. Now by going here, you can see the interfacial tension. Now we will go to the statistical part. Here you can get your approximate 72.8, which is in good agreement with the available value. Reportedly, people found 72.8 as the interfacial tension for the air-water interface. This is how the base standard deviation is calculated. This is how we measured the interfacial tension of the air-water interface using the pen and drop technique. Thank you for joining us.